Hi everyone, so I'm Zeynep and I'm presenting my engineering senior design work, which is in vitro characterization of DNA-bound self-amplifying RNA and its controllable, potentially therapeutic application. My teammate is Stella and we're working with Dr. Wilson Wong and Professor of Biomedical Engineering. So how many of you have ever been vaccinated? Show of hands, right. How many of you did you get booster shots for any specific vaccine before? Right. So most of us got vaccinated with the COVID-19 mRNA vaccines. And mRNA actually has a very short half-life, which just means that it degrades really quickly. So the mRNA vaccines that we got for COVID, but also most vaccines actually require booster shots to be effective and or high dosages, which actually creates a problem because like one shot is not enough. So you have to go back to the hospital to get another shot. So this creates like an accessibility and affordability issue. And the high dosages actually sometimes lead to unwanted safety problems. So what are some other problems with the current vaccine technologies? The live attenuated or viral vector vaccines, actually you deliver weakened germ or delivery with a modified virus, can actually have some safety concerns and side effects, but also, especially for immunocompromised individuals which have like a very weak immune system, and it might lead to like effectiveness concerns because of a pre-existing immunity already existent of the individual through like an infection before. And the subunit vaccines where you kind of deliver the specific piece of the germ is actually very time-consuming, complicated, and expensive to manufacture, which leads it to be a not versatile and not scalable tool. And in the case of like a new epidemic, pandemic, or like emergent diseases, variants, this might actually be a problem because it would take a lot of time for us to create a new vaccine. And it also requires booster shots, the same problem as before. And actually, some viral vector vaccines and mRNA vaccines include some of the early COVID-19 ones require ultra-cold conditions, which means ultra-low temperatures. And this need for special transport, handling, and storage actually leads most vaccines to be not accessible, especially in low-resource settings. In 2020, 3 billion people out of 7.8 billion people reported living in a place with insufficient temperature-controlled storage. Here is actually a picture of like the UNICEF trying to ship ultra-cold freezers to low-resource settings. So our solution is actually creating solutions to all these problems. It is genetically and thermally stable, so no need for ultra-cold storage and special handling. It's easy to manufacture and purify, so it's scalable and versatile. And it allows for amplified efficiency and longer-term effects through no booster shots or like high dosages. So backing off a little bit, inside the cells, DNA is the molecule coding for instructions to build and maintain a living thing, and then it gets transcribed into RNA, which is an intermediary molecule carrying instructions materials for subsequent processes. And then eventually this gets translated into proteins, which is the main performer molecule of biological processes. So our solution, which is DNA-bound self-amplifying RNA platforms, is us creating plasmids constructs, which means circular foreign DNA carrying instructions for desired processes, and like us launching in the form of DNA in mammalian cells. And then through transcription, this creates a specific type of RNA that has a self-replication machinery already incorporated in it. So it is able to self-sustain through an RNA replicating enzyme. So it kind of leads to amplified protein expression and a longer-term effect. And promoter is an important region that we're going to talk about later too, which is a DNA region upstream of a functional DNA fragment needed for making RNA. So again, all these great features, but interestingly, design principles are very understudied. So our goals for this project were characterizing these platforms through variations in design parameters and creating these platforms with additional features like a switch mechanism for enhanced safety control, which just means you like turning it on and on and off whenever you want, which kind of gives you the power to like account for the side effects. So obviously with the interest of time and biology just not liking you sometimes, we had to kind of like change the goals a bit. So we kind of focused on changes in promoters as design parameter variations, and instead of characterizing both immune response profiles and expression profiles, we only focused on expression profiles in vitro, which just means in lab setting. And in terms of the methods used, we kind of like take out and amplify the pieces that we want and put them together, fuse back in. And through a bacteria selection process with antibiotics, we actually select for the correct plasmids and then we verify. And then in cell experiments, we take these plasmids and we put them together with positively charged nanoparticles for them to like easily enter the cells. And upon translation, this kind of creates like a protein that is glowing, basically, which is like a fluorescent protein. When you like give a certain wavelength of light, it emits a certain wavelength of light, so you're able to see it as a specific color with the specific Citation 5 imaging tool. So in terms of the results, we're going to first look into the bright field microscopy. We had 3 groups, which is like only cells, which is a negative control group. And then we had 2 DNA siRNA systems with different promoters. And we can see here that in the bright field image, we have like similar cell densities for all of the groups. And then for the second column, we actually kind of added these DNA siRNA platforms with a control plasmid that was encoding for a red marker protein. So the fact that we see very similar red protein showing up in the second and third groups shows us that we had actually a similar efficiency in terms of adding the plasmids, which is what we wanted and expected. And in that similar efficiency transfection, us seeing a different expression profile for the 2 different groups of promoter DNA siRNAs actually proves the point that us doing these design parameter variations leading to different expression profiles for our platforms. So that was pretty cool to see. Current work, we're doing experiments with flow cytometer for a more discrete and quantitative measurements for expression profiles, and we're also, the plasmids for the drug-inducible switch system is already made, so we just need to make more experiments with plate reader to show the system turns on upon drug addition. Future directions, creating a more diverse library, and also doing the immune response characterization. Hopefully one day in vivo studies, which is within the living, and then through clinical application. This will allow for better vaccine and gene therapy delivery platforms, which means adaptable, safe, stable, more accessible, and long-term efficacious. In terms of the limitations, people who choose not to be vaccinated due to misinformation or mistrust, we think that we can actually convince these people through great and effective health dissemination campaigns, like an example here. But people who choose not to be vaccinated due to religious, political, and philosophical beliefs, and people who cannot be vaccinated due to severe allergic reactions are people that we cannot reach, unfortunately. So coming back to the COVID-19 example, what would happen if this technology was already implemented while COVID-19 was there? We would only get 1 shot and we would not need booster shots. It would be very easily manufactured and scaled, and it will be accessible for many more people because it's genetically and thermally stable. And it would be safe because you could turn the system off if there are side effects with like taking the drug switch system. So we would take quick and effective action with more lives saved. That's it. Thank you so much. Any questions? Yes.
Hi, great presentation, of course, loved it. I had a question actually about the no booster needed. Is that because of the promoter that was designed, or like, could you maybe just expand on that a little bit more? Like exactly
so
like why?
the reason why we need booster shots usually is the immune response effect is actually not that long term because kind of the antibodies like go away after a certain point of time, right? But with this system, through the self-amplifying RNA, which is able to self-replicate and like self-sustain, the RNA will be like staying around in the system. So the protein will, like the antigen itself will be present in the system so that this will lead to like the immune system to be active against it for a very long time. So we would not need actually booster shots. Yes.
This is great, Zeynep. I know this is a long way off, but from what you understand so far, what would you expect to be the biggest hurdle in developing this kind of vaccine?
Great question. So I would say currently, in the current landscape of things, Unfortunately, the vaccine work is not getting that much funding. So my concern would be like, this is a very new technology that's not established kind of at all. Like the DNA Lounge platforms are only present for very specific certain infection types, and that's it right now in the field. So kind of getting a grant approved for research on this and then getting an FDA approval would take a very long amount of time and funding and effort for it to be implemented. I think that is like the biggest concern as of right now. But
We could take one more question if anyone has any last ones. No? If not, great. Thank you so much.
thank you.